Using the assays presented here, the difference in phenotype among rotavirus strains can be evaluated when unique strains are isolated. Specifically, the phenotypic change of disinfectant-resistant strains of rotavirus is investigated. To achieve clear quantitative outputs using the plaque assay, it is critical to select an appropriate combination of rotavirus strain and cell line, in which the rotavirus is well adapted to the cell line. Demonstrating the procedure will be Syun-suke Kadoya, a doctoral student from my laboratory.
Remove a cryotube containing Ma104 cell lines from the liquid nitrogen container. Place the cryotube in a water bath at 37 degrees Celsius to thaw the cells. Add 1 milliliter of the cell suspension to 20 milliliters of the serum-containing medium in a T75 flask. Incubate the flask in an incubator at 37 degrees Celsius and 5% CO2 for 2 to 3 days. Once the cell monolayer reaches 80% confluency, remove the supernatant and wash the cells twice with 5 milliliters of 1-X-Delbecos PBS. Add 4 milliliters of 0.05% trypsin EDTA to the flask and incubate at 37 degrees Celsius for 5 minutes to detach the cells from the flask. Transfer the cell suspension to a 15 milliliter tube and centrifuge at 190 G for 5 minutes. Discard the supernatant and resuspend the pelleted cells in 1 milliliter of serum-containing medium. Then, dilute the resuspended cells 100-fold with the medium. Add 3 milliliters of the diluted cell suspension